Hi, I'm Tina. I'd like to talk to you about how to navigate the multiverse of skin conductance response quantification approaches. Skin conductance is one of the most abundantly used outcome measures in our field, and I'm not going to introduce the measurement here, but I'd like to introduce you to past me working with my first set of fear conditioning data during my PhD and wondering, how do I quantify the skin conductance response? What I did was that I had a look into the literature to find out how other people have been doing this and how I should do it. But this left me somewhat confused, and this quote describes my feelings very well, that there was a disconcerting diversity of electrodermal measurement techniques, which at best make it difficult to compare one set of results with another, and sometimes may cast even real doubt on the interpretation of the findings. And this quote is from way before I started my PhD. And I think much has changed with respect to our core recordings in the lab, but the problem seems to persist. And a decade after finishing my PhD, we did a systematic literature search to find out what kind of approaches are used in our field and how abundant they are. And what you can see is that trough-to-peak scoring seems to be most abundantly used, followed by baseline correction approaches and model-based approaches. Then we applied the different approaches that we identified here to two different data sets. So for both data sets, we had two individuals performing computer-assisted trough-to-peak scoring, ran the custom baseline correction script, performed LeaderLab CDA method, and also 4 different models in PSPM. What you can see here is the effect sizes for the CS discrimination during fear acquisition training as derived from the same set of skin conductance raw data, but with skin conductance responses being quantified through the different approaches that I introduced before. So to find out if these different approaches can be used interchangeably, we applied a replicability framework that defines a replication as when the effect size point estimate of the replication falls within the credible interval of the original study. Here, the reference approach, which in this example would be trough-to-peak rata 1. So applying this definition, the effect size point estimates here of rata 2, baseline correction, and LeaderLab would fall within the credible interval of our reference approach and would qualify as a replication, whereas none of the PSPM models would. For the mind sample, which differs in key procedural characteristics here, only the LeaderLab approach would not qualify as a replication. We also looked into trial-by-trial agreement across the different approaches that we used. And you can see that for the Hamburg sample, for the US and the CS Plus in black and red, agreement across the approaches, the trial-by-trial agreement is moderate to substantial and somewhat lower for the CS minus. For the mind sample, however, the agreement for CS plus and CS minus is mostly substantial to moderate, whereas agreement for the US is only poor to fair. And you can derive the disagreements from this pairwise trial-by-trial agreement between the approaches. So I will not go into detail here for time constraints, but you can pause the video and have a look. Finally, we looked into the agreement between the two trough-to-peak raters, which has been discussed in the field. And what you can see here is an individual response to a specific trial as scored by rater 1 and rater 2 connected with a single line. And what you can see is that both in the Hamburg and the mind sample, agreement is near perfect. So we seem to do quite a decent job in training our raters, and this is good news, I think. So let's discuss if the range of agreement between the approaches can be considered satisfactory and where to go from here. And I'd like to close with thanking my wonderful team, and particularly my collaborators that were involved in the results that I showed today. Thank you.